Hi guys. So we're back with our cellular respiration, and today our topic is going to be the stages. So let me share it up and get it in present mode, and we'll be good to go. Okay, so we're looking at cellular respiration, and specifically here we're looking at the stages of cellular respiration. So we have glycolysis, pyruvate oxidation, and the citric acid cycle, also known as the Krebs cycle. You'll notice they're the same color in this diagram because some resources always put them together as one stage. Some separate them out as two separate stages. So I just want you to be aware of that. And then the electron transport chain and chemiosmosis. So there are three key stages. Typically, that's how it's referred to. Sometimes one of the pyruvate oxidation is separated out, but in reality, it's still part of that stage most of the time. So we have glycolysis, pyruvate oxidation, and the citric acid cycle. Sometimes that's just in general referred to as Krebs cycle. And oxidative phosphorylation, which is the electron transport chain and chemiosmosis. So we're going to start with glycolysis. All living things perform glycolysis. If it's alive, it does glycolysis in some way, shape, or form. So this is the starting point of cellular respiration. This is where it always starts. So we're going to take that glucose molecule, and this is where we're going to start breaking it down. It occurs in the cytosol. That's one of the reasons that we can say all living things will perform this, because the cytosol or cytoplasm is something that's found in all living organisms. And the gist of glycolysis is it's going to split the glucose, which is a 6-carbon molecule, C6H12O6, into 2 pyruvates, which are 3-carbon molecules. Sometimes you'll also hear this referred to as pyruvic acid. They're the same thing. So pyruvate and pyruvic acid are the same thing. And so here you can see the process, starting with the glucose, breaking it apart. And the branch that you see going off this way would also be happening with the one off the other direction as well. They just showed it once. Do you have to know every single one of these little steps? Absolutely not. We focus on the big picture. Glucose goes in. We get 2 ATP, 2 NADH, and 2 waters, and 2 pyruvate molecules. But here's the thing you've got to remember when it comes to glycolysis. Think about that bowl of sugar. It just sits there until we get some sort of energy input. So there is an energy investment stage. The cell is actually going to use ATP to phosphorylate the compounds of glucose to start the process. And then we have the energy payoff stage, which is where we actually get the energy produced through the process. So there is a net energy yield per 1 glucose of 2 ATP. So in reality, we would make 4 ATP, but we have to put 2 in, which is why we end up with a net yield of just 2 ATP. 2 NADH. Okay, so summarize this process. In terms of the energy investment, we put 2 ATPs in, we get 2 ADP plus P out. The payoff, we get 4 ATP out. So we put 2 in, we get 4 out. You deposit you deposit 50 cents and you get $1 out. So you really only made 50 cents, but you still made 50 cents, right? And then our NADs, we get 2 NADHs. So net, we get 2 pyruvate, 2 waters, 2 ATP, because remember we had to use 2 to start, and 2 NADH. It's what we get out. Okay, so quick check. Where does glycolysis occur? The cytosol or the cytoplasm. What is produced through glycolysis? Pyruvate is what's actually produced. Yes, we still make some ATP. Yes, we make NADPH. What is the net production of ATP? The answer is 2 ATP. Remember, it was that idea of we put 50 cents into the machine and we get $1 out. So we made an additional 50 cents because the first 50 cents basically went to replace what we put in. Okay. So next we move to the pyruvate oxidation and the citric acid cycle. This is where it gets a little more unique and complicated. Not all living organisms can do this. Only organisms that have a mitochondria. So if oxygen is present, the pyruvate enters the mitochondria. So this happens in eukaryotic cells, and it only happens if there's oxygen present. Now you know why you actually have to inhale oxygen. You have to inhale it for this process. I know that sounds a little weird, but you have to inhale the oxygen for this specific process of cell respiration, where oxygen is present. The pyruvate is going to be oxidized into acetyl-CoA. So that's what is happening where it says pyruvate oxidation. Acetyl-CoA is used to make citrate in the citric acid cycle. And so acetyl-CoA, we're using a coenzyme. And remember, from our enzyme lectures, we talked about coenzymes, and coenzymes are based off of vitamins. So we're going to use one of those here to make this molecule. Then we have 2 carbon dioxides and 2 NADHs produced during the citric acid cycle. So let's look real quick. Okay, so here's our pyruvate. Here's our CoA. So we're going to make NADH and carbon dioxide from this process of changing pyruvate into acetyl-CoA. And since we had 2 pyruvates to start with, we end up with 2 NADHs and 2 CO2s. Okay, so like I said, these two, the pyruvate oxidation and the citric acid cycle, are typically clumped together as one step, but every so often you may find a source that separates them, and I just want to make sure you're aware of that. So now we're moving into the citric acid cycle portion. So it's also more commonly actually known as Krebs cycle, named after the gentleman who first described it. And it will occur in the mitochondrial matrix. So that central portion, inner portion of the mitochondria. We're going to take that acetyl-CoA that we made in the pyruvate oxidation, and we're going to turn it into citrate. That's where the name citric acid cycle comes from. In this process, we're going to release CO2, and we're going to make a little bit of ATP. And we're also going to transfer some electrons to NADH. Remember that energy molecule? We made some of that in our previous steps. And a new one named FADH2. It's going to be just like the others. It's an energy molecule, but ultimately we can't use it until we transfer the energy to ATP. And so here you have a very detailed diagram of the steps of Krebs cycle or citric acid cycle. Do you need to know all of these steps? No. Not unless later on in life you are going to be studying the citric acid cycle in particular as like your main research area of focus, then you would need to know it all. So we're going to summarize the pathway based on one single glucose molecule. So remember, we already broke our glucose molecule down into 2 pyruvates, which then we turned into 2 acetyl-CoAs. So what goes into the citric acid cycle are our 2 acetyl-CoA. And it's going to go around, go through the process. And for one glucose molecule, we are going to come out with 2 ATPs. So we made 2 ATPs in glycolysis, and we have 2 ATPs here. So now we have a total of 4 ATPs. We're going to have 6 NADH, 4 CO2, and 2 FADH2. So we've got a lot of these NADH and FADH2s that we're going to have to exchange to make ATPs. Okay, quick check. Where does the citric acid cycle occur? In the mitochondrial matrix, in that center portion of the mitochondria. What is the net production of ATP in the citric acid cycle? We make a total of 2 ATP in the citric acid cycle. Okay, so we're moving on to the very last step, which is known as oxidative phosphorylation. This is the electron transport and chemiosmosis. So oxidative phosphorylation consists of the electron transport chain, which is a giant hot potato going from one protein to another, and chemiosmosis, where we're actually going to do the conversion to make that ATP. So here is a pictorial that kind of shows you the process. And if you notice, just looking at the process, here's our NADH. It's going to come drop off and we get those hydrogens floating. FADH2 is going to come drop off hydrogens. These hydrogens then are going to get moved as we're passing the electron. This orangey line here is the electrons. As we're passing those electrons, we're going to move the hydrogens. So we got a lot of hydrogens on one side of the membrane and not as many on the other. Then they're going to move through this molecule, our ATP synthase. And through that process, we make ATP, taking that energy and then that hydrogen. Anybody have a guess what happens to the hydrogens? If you hadn't noticed, it's right here. So we're going to take the hydrogens, the electrons, and that oxygen that we had to inhale to make this whole process possible, and it makes water. Okay, so the electron transport is located in the inner membrane of the mitochondria, so that folded mitochondrial membrane. We have a collection of proteins that make up the electron transport chain. And as the electron falls through those proteins, right? As we're going hot potato passing through the proteins, we're going to kind of alternate between a reduced accepting electrons and oxidizing donating electron state. So obviously when we pass it, it has to accept the electron, but then it's going to toss it on, so it's going to donate it. The cristae increase the surface area. So remember when we looked at that membrane, it had all these little like folds in it. Those were the cristae. And the idea is that they're going to increase the surface area so we can have more of this reaction occurring. That makes sense, right? This process itself, the electron transport chain part, doesn't actually produce the ATP directly. It basically sets the stage. So it's managing the release of the energy, creating all these little steps or falls for the electron, but it's not actually creating the ATP. The final acceptor is our oxygen. Remember, we have made this decision that we had to have oxygen present to go this route through the mitochondria to make our ATP. And so far, we've only made 4 ATPs. So each oxygen is going to pair with 2 hydrogens and 2 electrons to form water. And so that's what's happening with the electron transport chain. But one of the other big things and one of the most important functions is it actually creates a proton or hydrogen ion gradient across the membrane. So we have more hydrogens on one side than on the other. So the proteins are going to actually shuttle the electrons. They're going to pump the hydrogen ions into the intermembrane space between the two membranes. They use the exergonic flow of electrons. Remember, exergonic giving off energy from NADH and FADH2 to do this. This gradient powers chemiosmosis because we've got to have a gradient to have osmosis of any sort. We learned about that. So we're going to use those hydrogen ions to actually power this system. And so that's where our ATP synthase is going to come into play. For chemiosmosis, we're going to use that ATP synthase enzyme to actually make the ATP. And so notice our gradient, we've got lots more hydrogens on one side than the other, and we're going to move them through that ATP synthase. We're going to use the energy from those hydrogen ion gradient across that membrane. The hydrogens flow down the gradient from high concentration to low, we're used to that, through the ATP synthase. And it's kind of like a little rotor or a little spinning structure. When the hydrogen ion binds, the rotor spins. And it activates catalytic sites so that ADP plus P becomes ATP. It's kind of like how we generate electricity, if you think about it, with like a hydroelectric dam, is they actually have these rotors that are spinning due to the water movement. Well, we have a rotor that's spinning due to the hydrogen movement. And that spinning is what actually makes it possible to create the energy. So this, so far it seems like an awful lot of work, and what we've gotten 4 ATPs so far. This process makes somewhere around 26 to 28 ATP per glucose. Sometimes you'll even see numbers as high as 34. It varies. But remember, we always lose some aspect of energy whenever we're transforming it. So we went from, we did all this work and made 4 to all of a sudden add that 4 plus this other 28. So we're talking like 32, 34, 30. That's still a lot more ATPs. Quick check. How is a proton gradient formed across the inner mitochondrial membrane? So the answer is that the exergonic flow, the energy releasing flow of electrons from NADH and FADH2 powers the complexes in the electron transport chain to pump hydrogen ions into the inner membrane. That's what makes this gradient. What is the final electron acceptor in the electron transport chain? This should be an easy one. Oxygen. That's the whole reason we were able to do this process, was because of oxygen. How does ATP synthase obtain energy to convert ADP plus P to ATP? And that's from that proton gradient. As it moves through the ATP synthase, it causes that little rotor to spin, which allows for the energy conversion. So to summarize, Glycolysis, we put in 1 glucose, we get out 2 pyruvate, 2 ATP, 2 NADH. Pyruvate oxidation, we put in the 2 pyruvate, we get 2 acetyl-CoAs, 2 carbon dioxides, 2 NADHs. We take those acetyl-CoAs and put them in the citric acid cycle. We get 4 CO2, 2 ATP, 6 NADH, and 2 FADH. Now, let's stop for a real quick moment and think. That carbon dioxide, we know carbon dioxide is considered toxic in large amounts in the body. What do we do with the carbon dioxide? Hopefully you picked up that that's what we exhale. So the carbon dioxide that we are exhaling via our lungs actually comes from cellular respiration. Okay, and then oxidative phosphorylation, that's that electron transport chain concept. We take those 10 NADHs and 2 FADHs, 2s that we made in all these other steps, and we're going to exchange them to get 26 to 28 ATP. So we get a total of somewhere around 30 to 32 ATP, really. Like I said, it's one of those that you'll see higher numbers, and it is possible, but this is actually more realistic because we do lose a lot of the energy off as heat. So hopefully that helps you understanding the steps for cellular respiration. Bye.